Hello and welcome to another BYU-181 video on DNA replication and the Meselson and Stahl experiment. This is something that confuses students, how it worked, what the purpose of it was, what it explains, and especially the homework that is to do with this problem. So to do with this experiment, sorry. I hope this helps. To begin with, we're going to talk about three theories. There were three central theories in the 50s that different scientists had concerning how DNA replicates. There was the conservative model, the semi-conservative, and dispersive. I'll explain these three with just simple pictures. The conservative model, or theory, or hypothesis, whatever you want to call it, on how DNA replicates involved the idea that here's a DNA strand and it would go through the process of replication and you'd be left with the original strand would be the same, so obviously this matches with this, they're the same, and that strand would be used to make an entirely new double helix strand of DNA. That's how one of the ways, the conservative model, and how they believed that DNA replicated. Obviously we know this is not true, and we'll get there. The semi-conservative model, similar. So here's our original double-stranded DNA, and I might as well draw this one. I got blue on. Semi-conservative model believes that the original strand here, it splits into two, and the daughter strand is replicated like this, represented by the red strand. So this is the template, and both strands are unwound and copied, and newly synthesized DNA is made. So we know that this one's true. Semi-conservative. Dispersive is a little weird. I'm just going to draw these quickly. So it's believed that kind of both strands had a bit of a shake-up. So then, yeah, let's try that again. There we go. So both strands had a bit of a mix-up. So this DNA unwinds, and then when the replication occurs, the newly synthesized, some of the original DNA of both strands is used, but then new DNA is added in to, I guess, increase the quantity enough to have two new double helices, or whatever. Obviously this one's ridiculous. But back in the 50s, the scientists, these were their ideas, and they honestly didn't know which one was correct. And Meselson and Stahl did a fantastic experiment to prove that semi-conservative was the correct mode of replication. So how did they do it? Well, with the use of nitrogen-15 and nitrogen-14 radioactive isotopes of nitrogen. We know that in DNA there are nitrogenous bases, plenty of them. There's lots of nitrogen in nitrogenous bases, and so being in a nitrogen-15 solution, replication is going to essentially infect those nitrogenous bases with the radioactive nitrogen, allowing you to see it. So here's our E. coli, and what they did Meselson Stahl put the E. coli in this nitrogen-15 solution, and over time, with the replication, the DNA essentially became infected with the nitrogen-15. So eventually it looked like this, and then with this tagged DNA, they collected it and they centrifuged it, or they spun it really fast. This test tube they filled with cesium chloride solution. These ions, cesium chloride ions, separate, causing an electrochemical gradient as positive and negative charges, and that attracts the DNA because we know the DNA is negatively charged. I put the positive and negative the wrong way around, never mind. The nitrogen-15 is dense, so it goes lower, and it allows you to see the DNA in this region of the test tube. Then they get that DNA, they put it back into an E. coli, into the E. coli population, and then they put that back into a nitrogen-14 solution instead, and allowed it to replicate. After each replication cycle, for 4 generations, they centrifuged it, and allowing them to see how the DNA changed. So I'm going to show you what they did. So I apologize, it's going to take some time for me to explain this and show you the results. So I'll just draw a diagram that's similar to what's in the book. Here's our test tube, the ultracentrifuge, and there's the cesium chloride solution, different charges, and then I just draw these different boxes. So here's generation 0, 1, 2, 3, and 4. We'll ignore this last box. So for this one, for this generation 0, this is the pure nitrogen-15. They centrifuged, and they saw that it was in this region. So it was more dense. And I suppose that this shows the density and shows the quantity of nitrogen-15 or nitrogen-14 in the DNA. Density is increasing as we go down. So this here is pure nitrogen-15. I hope you can see that. Sorry if you cannot. And then they put that nitrogen-15. We know they put that into the DNA, the E. coli. They transferred it into a nitrogen-14 solution, allowed it to replicate, and then they centrifuged again, and there was a slightly less dense band appear in this test tube, and so that's a little strange, and I'm going to call this the hybrid DNA, hybrid between nitrogen-15 and nitrogen-14. Maybe a little bit of orange in there, I don't know, because I'm going to use red to represent the nitrogen-14. And so then they replicated it again, and in terms of quantity, this was 100% nitrogen-15, and then this was 100% hybrid. After the next replication, there was this split, and so we went to 50-50. 50% was the pure nitrogen-14, and then 50% was this hybrid again, this hybrid DNA molecule. The quantity was splitting, and so we're slowly increasing in the nitrogen-14 quantity. And then after another replication cycle, this band was getting bigger and this one's getting smaller. Sorry, the color's slightly changing as we're going along. I have a bad way of selecting the colors. So now we're 75% pure nitrogen 14 and 25% pure nitrogen, hybrid nitrogen 15 and nitrogen 14. And then after the fourth round of replication, let's do this last one. There's even more, huge quantity. It looks like there's more to start off with. Sorry, it's been my bad drawing, my bad representation. Focus on the numbers. The diagram's just kind of helping give you a visual. Focus on the percentages. It's 88% of the DNA was pure nitrogen 14, and then a pathetic 12% was now the hybrid. So that as soon as one replication cycle happened right here, we went from 100% pure nitrogen 15 to no nitrogen 15 whatsoever, and there being this hybrid. And so, let's change the color. We went from being like this, instead of me using just words, I'll use these pictures, and so we went from this essentially to this completely. And then there was still some of the hybrid we had. So now you know visually what I'm saying with pure nitrogen 15, hybrid here and here, and pure nitrogen 14 DNA. So yeah, I really hope that's making sense. If not, pause the video, go back, watch it again, try and understand what's going on here, and use these diagrams and this representation of the results of the Meselson Stahl experiment should really help you see what was happening. All the nitrogen 15, there was lots of it, lots of the E. coli. All of their DNA looked like this. But then over time, after one replication cycle, it changed to this hybrid. And then that split and went 50% pure and 50% hybrid. So what happens here, I guess if I can draw it, this hybrid split, and you have the blue bit separate and the red bit, or the nitrogen 15 and the nitrogen 14 molecules separate, and then they replicate, and they replicate in the nitrogen 14 solution. So the daughter strand is going to be the nitrogen 14, and so now we've gone to 50-50, like so, right here, 50-50, nitrogen 14, nitrogen and hybrid. So yeah, make sure you watch this a few times if you need to understand it. And now what I'm going to do is show you basically how to solve your homework. In your homework, you're asked to take it from this generation 4 and this DNA, you take that E. coli, you take this DNA, and you transfer it into a back into a nitrogen 15 solution. And you're supposed to show what happens after, I believe, 2 generations. I'm going to show you what happens after 1 generation, and then hopefully from that you can figure out what to do for the rest of the homework. So we're going to take this DNA and do generation 5. So, sorry this is a long video, I promise it's almost over. So we've got this DNA is 88% pure nitrogen 14. Nitrogen 14. What's going to happen to that? Of course, the replication will occur, and we're going to have the separation of the 2 strands, and then the replication will occur, and we'll have 100% hybrid, right here, everything. So 100% hybrid, well, it's only still 88%, right, because there's this 12% we haven't accounted for. So if we were to draw it, I'm not going to draw it yet, bear that in mind, keep that in mind, we'll put that on the back burner. Now let's focus on this current 12% hybrid, what's going to happen to it. So we've got this, oh whoops, let's get the red. This is our hybrid DNA in this part of the E. coli, in this colony of E. coli, and so that's going to separate, and we're going to have the molecule, the DNA molecule separate. If the semi-conservative model be true, and which it is from this data, the molecules separate, and they're the parent strands and the daughter strands. Because it's nitrogen 15, is going to be like this. So now we have a 50-50 split, and so we went from being 12% hybrid. 12% hybrid is now 50-50, so the 6% hybrid and the 6% pure. So now 88% plus 6% hybrid. So we can draw on our diagram right here. This 6% hybrid and this 88% hybrid. Going to be about 94% hybrid DNA, and then the other remaining 6% right here is going to be back in line with the, well, it's nitrogen 15, so it's going to be lower, it's more dense. Right. So there's this little band here of the pure nitrogen 15, and then the rest is the hybrid. And we know that from the semi-conservative model. That's what the 5th generation will look like. It'll be 94% hybrid and 6% pure. Now your job is to figure out the rest. Good luck, and thank you for watching.